This is a very, very special afternoon and evening, and tomorrow as well. We, of course, have a very special guest, Professor Harlan Varmus, who is a cancer specialist and a Nobel Laureate. So we'll hear more details about his life and work when he's introduced as a speaker. But I just want to acknowledge that he is the C.V. Raman Professor at the Indian Academy of Sciences. His memoir, The Art and Politics of Science, is something that we should all be reading if you haven't already started it. With that, let me invite our new Vice Chancellor, who has just joined Ashoka, Professor Shomak Roy Chaudhary, to give the opening and welcome remarks. Professor Chaudhary.
It's wonderful that one can start off the year with such a bang, with a couple of events centered around Professor Harold Varmus, who's visiting us. And we've had some really, really interesting conversations over the last day and a half. Also, what interests me more is that the topic of today's talk, of course, as things will unfold very soon, is so much consistent with what we are doing here in Ashoka, bringing together art, politics, science, and the different disciplines of knowledge and experience together in both our academic programs and everything that goes with it. And I can't think of a better way of starting this year with something like this. So welcome to you, welcome to Professor Varmus, and of course to introduce and say some initial remarks for before the talk is, of course, with us is Professor K. Vijayaraghavan, who also, of course, is a very illustrious scientist. Until very recently was a principal scientific advisor to the Prime Minister of the Government of India, and also is a very, very distinguished biologist himself, has a long association with the speaker today, and is now the chairman of the Science Advisory Board to Ashoka University. And in that capacity, of course, together, we're trying to see how science and associated subjects are brought into the Ashoka oeuvre, as it were. So thank you very much again, and welcome first to Vijay and then to Harold.
Thank you very much, Shomak. It's a great pleasure to be here at Ashoka. There is a great danger in introducing Harold Varmus because that can take up much of the time for the talk, and then we'll have time only for questions. But I'll try and be brief. I've also got a few slides which you can see. They have nothing to do with what I'm introducing, but they're just for fun. And Harold can see the slides here. So for several decades, Harold Varmus has advanced fundamental scientific knowledge at the intersection of virology, oncology, genetics, both as a researcher and as a science administrator. And with his collaborator, longtime collaborator, Michael Bishop, Varmus developed a new theory of the origin of cancer, which holds that the disease arises from mutations in certain of our normal genes, interacting with the environment, and so on. In his acceptance speech at the Nobel ceremony in December 1989, Varmus, a former graduate student in literature, compared cancer to Grendel in the epic heroic poem Beowulf, and said, and quoted, "In our adventures, we have seen our monster more clearly and described his scales and fangs in new ways, ways that reveal a cancer cell to be like Grendel, a distorted version of our normal cells." Harold Varmus was born on December 18, 1939 in Oceanside, New York. And like every Indian parent, his father urged him to go to medical school. And Varmus obliged by taking pre-med courses during his undergraduate studies at Amherst College. Nevertheless, he found his first intellectual passion not in medicine, but in English literature and in writing, earning an AB degree, magna cum laude in 1961, with a thesis on the novels of Charles Dickens and serving as editor of the campus newspaper. He considered an academic career in literature and even earned an MA degree in English from Harvard in 1962 with a focus on Anglo-Saxon and metaphysical poetry. However, Amherst friends who had chosen medicine seemed more fulfilled and excited about studying matters with immediate relevance to basic human needs. And so Varmus went on to medical school at the Columbia College of Physicians and Surgeons in 1962. He was rejected at Harvard, and initially with an interest in psychology and later in internal medicine. He received his MD in 1966, which included a 3-month internship near Bareilly at a mission hospital with also interesting consequences on his gut. During a backpacking trip to California, Harold Varmus met with Michael Bishop and his colleagues, and they had begun to study retroviruses to detect cancer-causing genes. And this brief conversation led to a long collaboration. And as Michael Bishop recalled, the relationship evolved rapidly to one of equals, their bond formed not just by shared fascination with cancer viruses, but our mutual love of words and language. In a collaboration which was unusual in an era of increasing competition in science, Bishop and Varmus together directed a research group. I forgot to change my slides, by the way. Yeah. Bishop and Varmus together directed a research group during the 1970s and early 80s, and that eventually numbered more than 24 and made all of their major discoveries as a team. Varmus and Bishop's discovery, which you will hear more about tomorrow, yielded a better understanding of how retroviruses change the fundamental behavior of cells and brought scientists closer to a unified theory of cancer. And in 1989, Varmus and Bishop shared the Nobel Prize in Physiology and Medicine for the discovery of the cellular origin of retroviral oncogenes. Now, till then, Harold had no experience in administration as head of a department even. But in 1993, President Clinton of the United States nominated Varmus as director of the National Institutes of Health, the nation's premier research and funding institutions in the biomedical sciences. And he was the first Nobel laureate to lead the NIH and strengthened its commitment to basic research while balancing demands of advocacy groups for more targeted research on particular diseases, most notably AIDS. At the start of his term, he said he would stay for 6 years, and after 6 years, he left to become the president of the Memorial Sloan Kettering Cancer Center in New York, and served as 10 years of the head of that institution. He expanded the center's basic research educational programs and strengthened connections between lab and clinical staff, and built stronger bridges with Sloan Kettering and Weill Cornell Medical School and the Rockefeller. In 2010, Varmus was called back, and this time as director of the National Cancer Institute, which is part of the NIH, and he had greater flexibility in focusing on science in that role and played a very important role in cancer programs, about which you'll hear again. Since 1999, he led a program to make publications and scientific publishing more accessible. That led to the creation of the Public Library of Science, and that's been really a major landmark in our ability to access the biomedical literature. He's the author of more than 300 scientific papers, 5 books, and as you heard, The Art and Politics of Science is a very popular book which tells us about his life as an administrator and as a scientist. Varmus has been married to journalist and book critic Constance Casey since 1969. They have 2 sons, Jacob and Christopher, and Harold left the NCI in 2015 to go back to New York, where he has a small research lab and still teaches. So that's him. He's an exercise fanatic. That's in Gujarat and Delhi, actually, after a Gujarat meeting of several Nobel laureates. That's Varmus and Bishop. Michael Bishop postponed the press conference at their Nobel announcement because he wanted to watch a baseball game. So this is a cartoon representing that. Thank you very much. And with that, Harold Varmus.
Okay, well, thank you very much. First of all, I want to express my gratitude to the Indian Academy of Sciences, which invited me over 3 years ago to give the Raman lectures. And there was a slight intervention here due to a coronavirus that kept me from coming to India. And I then want to thank Prof. Majumdar, who was then the president of the Academy, who has borne with me for the last 3 years as I repeatedly postponed my appearance in India. And Prof. has had to suffer the indignities of scheduling and rescheduling these lectures several times. This is the first of several lectures I'll give over the next 10 days or so, and it's intended to provide some introduction, even slightly more lengthy than Vijay's introduction of me, and you'll hear that there are some resonances, some compatibilities between what the two of us had to say. And tomorrow I'll be giving a talk that is much more scientific in content. Today I'm mainly going to focus on my life history as a way of introducing myself to you, but also a way of illustrating how life doesn't spin itself out in a totally orderly fashion, and often evolves in a way that I think is consonant with many of the principles of what led to the establishment of Ashoka University, namely a recognition that the humanities and the sciences and the arts intersect in many interesting and important ways, that lives are shaped by a variety of influences, that change can always occur, that one may think you're starting in a straight line toward a target and be deterred into a different direction. There are a few caveats about today's lecture. First, there's going to be very little science. Actually, I'm grateful to Vijay for showing more about my science than I'm going to say myself in today's lecture. That's very helpful to me. I want to say that I have to recognize that the story I'm going to tell you has got a pretty happy set of circumstances and a pretty happy outcome. And I am aware of the high level of privilege that I have, beginning with the fact that my ancestors came to America before they were killed by the Nazis. They were Jewish immigrants to America. And I lived a life of relative prosperity and happiness in a world that I recognize is beset by lots of problems, many of which I've tried to help address, but haven't experienced myself firsthand. I also recognize that I'm looking at a small slice of life, one that is informed by those levels of privilege and the good circumstances in which I've been existing on this planet. I also want to express my appreciation to Ashoka for having me here. I recognize that there are a few students who aren't on campus these days, but I'm getting some sense of this exciting new opportunity that's been created by your founders and the great faculty that have been recruited here. And it's a pleasure to be spending time with them. So I think it's no secret that this lecture is entitled with the title of my book, and I'm glad to see that at least someone down there in the second row, hi, how are you? Thanks for buying it, have bought and perhaps even read part of this book. My goal here is reflected in the title, to tell you something about how science is influenced by both art and politics. I also would point out to those of you who have some interest in hearing more than what either I or Vijay had to say about this book by pointing out, if you can see it at the bottom, that this book is consistent with my own principles, now fully available online at no cost, just by going to what's called the NIH bookshelf and downloading a copy for yourself with no money flowing into my own hands. So what I'd like to do is outlined here in today's menu. I'll give you a few things about my childhood and adolescence that indicate my style. Then to tell you about three major topics that are featured in this book: how I became a scientist, what I did, something about what life was like being a scientist without getting into the boring details, and then telling you something about how my life shifted quite a lot from practicing science obsessively to being a science administrator although I don't like the word administrator, a leader in the way science works, at least in the US, helping others to make discoveries and trying to encourage more science and more discovery in poorer parts of the world. And I hope there'll be time for questions, store them up for the end. And I know we have an obligatory panel discussion here, which we'll carry out at the end shortly before dinner. So something first about my background. My grandparents, all four of them, came from Europe, from Eastern Europe, sometime between 1880 and 1910, and under various circumstances that I don't have time to describe, escaped persecution and poverty to arrive on the eastern coast of the U.S. One of the remarkable features of immigration to America, at least in those times, is that my parents, who were born some years after their parents arrived here, were able to go to prestigious colleges. My father was a Harvard undergraduate and went to medical school at Tufts. My mother was a college student at Wellesley College. They both became professionals. My father, a general practitioner of medicine, my mother, a psychiatric social worker. And the only interference with their lives early in my life was when my father was serving in the armed forces during World War II as a physician at a Florida air base. That picture shows us living not far from Orlando, Florida, while he was doing that service. After that, we moved back to Long Island, where he was the Jones Beach State Park physician, in addition to being a family physician. And I spent a lot of blissful afternoons at Jones Beach State Park, and was assumed as the male child of a Jewish physician to be destined to be a doctor myself. And I went through public high school assuming that life would lead to a pre-med undergraduate curriculum, medical school, and medical practice. But as Vijay has indicated, I went to a liberal arts college, Amherst College, which I like to think, based on what I have gotten from my friend Pravetti here, is one of the models for your college, Amherst College, and a place founded in 1821, mere 200 years or so before Ashoka, where the curriculum was very general. Every undergraduate took the same 2 years of courses before becoming a major in some specific field. And during that early time, I learned that not only did I like to read, but I was genuinely deeply interested in English literature. Saw that there was theory as well as fun to be found in the study of literature. Indeed, a kind of science was imposed on my thinking about literature through the writings of I.A. Richards, who tried to stress the importance of looking at the words on the page. That's evidence, as opposed to receiving accepted opinions of what writers were like and what they might have thought. That was a way of thinking that science and the humanities were not so different. And along the way, I fell in love with the writings of Charles Dickens, shown on your left. I wonder if the lights could come down in the front, because the pictures are not as effective as they might be if the lights were a little dimmer. And I wrote an undergraduate thesis about Charles Dickens and how he operated as a narrator, trying to control characters who were sometimes good, but too weak to overcome evil. And I decided that even though I was still doing a pre-med curriculum, thank you. The best thing would be yeah, that's good, thank you. Even though I was still doing a pre-med curriculum, I began to consider the idea of going off to graduate school, which I did. And as you've heard, learned Anglo-Saxon and read Beowulf and metaphysical poetry, but was a bit dissatisfied with my future as a professor of literature, and decided instead to consider going to medical school, and did again reapply to a few schools, and was fortunately accepted at at Columbia Medical School. As I reflect on those few years, which were obviously important for my future, since I didn't know what my future was, and I was playing around with a lot of ideas about what I ought to do to figure out what my course in life ought to be, I realized that in retrospect, that over the next 30 years or so, the next 10 years or so, that a number of things happened that influenced my decision to eventually become a scientist. At this point, 1961, when I graduated from college, I really was very unsure about what I should be doing with myself. I really could not have told you where I was going to end up doing my life's work. But I realized in retrospect that during the next 10 years, up until about 1970, that at least four major things happened that influenced me in the direction of doing science. The first, curiously, occurred just after my graduation from school, when I was accompanied a classmate whose name is Arthur Landy, who became a prominent molecular biologist himself. He won the Amherst College Biology Prize, and the prize that year was to attend an international scientific meeting. And he made an interesting choice to go to the first international meeting in biology that had occurred in the Soviet Union for about 30 years. And those of you who know some history of science, I imagine Vijay is one of them, were very excited about the idea of going to the Soviet Union, where the charlatan Lysenko had been dominating science there under the influence of Joseph Stalin for many years. And with Khrushchev having taken over the country, he encouraged the idea of having an international meeting to get caught up on what was happening in biochemistry and genetics. And to everyone's surprise, at that meeting, Marshall Nurenberg, a famous scientist at the NIH, announced the first inklings of how the genetic code worked, learning how the four letters that compose DNA, which I assume everyone here is familiar with, might be a means of teaching a cell how to make proteins from based on a genetic script that was encoded in DNA. And that I have to admit that I was not in the room when Nurenberg made that announcement, but I was sharing a dormitory room at Moscow State with my friend Art Landy. And he came back one night after I had been spending the day touring the Tretyakov Art Museum to tell me that the genetic code had been announced and everybody was terribly excited. This was only a few years after Watson and Crick had figured out that DNA was a potential coding device for genetic information, but nobody really knew how that information could get out and be read by machinery that would make proteins. But it wasn't too long thereafter that it was figured out that there was an intermediary called RNA that carried the information from the nucleus, DNA, into ribosomes that were able to translate genetic information into the formation of cells' repertoire of proteins. So I realized, even though I was at that point heading off to graduate school in literature, that this was a moment of that was inducing a great deal of passion in an audience, a kind of passion that I didn't think I would encounter among fellow scholars in the literary tradition. So at this point, I had transferred to medical school and at Columbia, and while much of medical school was frankly rather boring, I was in a great cultural center, New York City, and spending my time with some very lively people. This is a picture of a bunch of us in the classroom during my second year of medical school. I may be recognizable over on your left. Sitting next to me is one of my best friends from medical school, Robert Lefkowitz, who is also a Nobel laureate for his work on cell signaling. So fairly high density of future Nobel laureates in that medical school class. And although we're seen here smiling and having a good time with our instructor, we did learn some serious things. And one of the things that really impressed me during medical school was the way in which certain diseases like anemias were being understood at the molecular level, that sickle cell disease and thalassemia and a number of other hematological disorders could be traced to single changes in the genetic code. And that seemed like a very bright prospect for understanding disease at a more fundamental level. So even at this point, when my mind was directed toward a career in psychiatry or internal medicine, I realized that what was going to be an exciting future for medicine was more likely to lie in the laboratory, where information in our DNA was going to be a compelling component of understanding the origin of disease. Now, during medical school, we were offered a chance to spend a few months doing some kind of elective activity. And because of my various interests in other cultures and living abroad, I chose to come to India for a few months. And as you've heard, I went to a town which probably not too many of you have actually gone to, Bareilly, India, in northern UP, to work in a mission hospital. I confess I'm not particularly religious, and I only ended up at a mission hospital because I was told that was a place that I could go with a fellowship I had received from a drug company to spend a few months working with Indian doctors and some missionary doctors as well. Clara Swain Mission is actually an interesting place. It's a hospital founded in the middle of the 19th century. It's the oldest Methodist mission hospital in India. It had a very interesting structure because it was kept cool by air circulated up from tunnels that had been dug deep under the hospital. So even on the most grueling days of spring in Uttar Pradesh, life was fairly cool in those corridors. And I had a very interesting time learning about the treatment of tuberculosis and leprosy in particular at that hospital. In addition, I learned some terrific things about life in India. And because of some connections with friends of mine at Amherst who were studying the history of Bengal, I met people who were friends of Satyajit Ray and indeed spent an evening with him at his house. I arrived in Calcutta in 1966 at a time when labor unions and Communist Party members were overturning buses and had induced the arrival of troops on the road from Dum Dum Airport. And this picture showing what life looked like to me as I arrived as an innocent in Calcutta in 1966. So it was a multifaceted and interesting time for me that expanded my horizons dramatically and influenced, as you'll hear, my approach to spreading science and medicine throughout the world. The final phase of these several events that influenced me in the direction of medical science and global health was the involvement of the U.S. in a war in Vietnam, a war that I suspect many of you have heard about, but it was one that engaged America in a rather pernicious way, in my view, but required, because many physicians were opposed to the war, it resulted in the passage of a bill that required that all male medical school graduates serve time in national service, and that generally meant serving in the war in Vietnam. I was not alone, as you'll see, in being one of those who sought to comply with the law without having to go to jail or go to Canada by working in the public health service. And despite my almost complete lack of any laboratory experience, I was fortunate to be signed up to work at the NIH under a man slightly older than I was at the time, Ira Paston, who had experience working on as a physician working on the thyroid gland, and had recently developed an interest in a chemical messenger called cyclic AMP that had been discovered in bacteria, its function unknown. He made, as I'll describe in a little more detail tomorrow, a major discovery about how that messenger molecule from mammalian cells, cyclic AMP, could have an effect on the behavior of genes in bacteria. And that had a strong influence on my, as I'll outline in just a moment, on my thinking about approaches to disease. So this was an alternative to military service, very important from my well-being, both mental and physical. And it also served to introduce me to the remarkable things that were happening and as a result of the rise of some of the tenets of molecular biology, the understanding of genetic codes, the understanding of how genetic information could be converted into proteins, and how this could lead to an understanding of how genes are regulated and how cells receive and transmit signals. I was certainly not alone in choosing the NIH as a vehicle for escaping military service. Here's a picture of my cohort at the NIH in 1968 or 9. As you can see, there's a bit of a gender imbalance here. I think there's one woman somewhere buried in this picture. The rest of the group were male medical school graduates who were doing their national service as members of the public health service by working at the NIH. Now, I learned a lot of things in my 2 years at the NIH. And one of the first things I learned is that nothing is as pleasing to somebody who's starting to do some science as having a powerful assay, a way to measure things. And many other scientists have commented on the power of an assay. For me, it was the most important day of my scientific career was probably the day when I knew that by having massaged some molecules in a certain way, I was able to tell you just how many molecules of messenger RNA from a gene called the LAC gene were present in a bacterial cell, and that I could use a scintillation counter to detect radioactivity that represented those molecules, and that I could measure how a gene was being regulated and when it was being regulated in a very simple and powerful and reliable way. The second thing I was learning was that using very simple systems, bacteria like E. coli, to study the regulation of a single gene could be a very useful model for thinking about how genes are regulated in more complex organisms like human beings. And then, of course, as I tried to answer some simple questions that I'll talk about more tomorrow, I learned that the good answer to a good question was what led to more questions. And that's how scientific careers develop. You find something you would like to answer, you get an answer, inevitably that answer drives additional questions that are fun to try to answer. Then I learned how important it was to talk to other people, that one of the pleasures of doing science is not just getting an answer, because who are you going to brag about that answer to? And then you learn that scientific gossip is not just about other things in life, but also involve telling people what you've done, why you've done it, and why the answer is interesting, and that a life without colleagues to gossip to would be a very unsatisfying life in science. And then I learned something that was even ultimately more profound for me, that these molecular methods that I was applying to simple bacterial systems could conceivably be applied to understanding human diseases. I had begun to learn that lesson as a medical student, thinking about anemias, but even a disease as complex as cancer, a disease that at that point was killing my own mother, was something that influenced my next step, which was to think about ways in which these molecular methods could be studied in animal systems to try to understand complex diseases. And indeed, at that point, I decided that I would find a place consistent with my other cultural interests, such as trout fishing and mountain climbing, would be best pursued by doing some additional postdoctoral work on what I thought was the best approach to cancer at that time through the use of viruses that cause cancers in animals. And so at that point, I moved out to University of California, San Francisco, to think about how I could try to understand cancer more profoundly by studying an important type of cancer virus that I'll, again, talk about in more detail tomorrow, the Rous sarcoma virus, a virus of chickens that could induce a kind of cancer called a sarcoma. So for the next, turned out 23 years, I was pretty heavily focused on this issue of what certain viruses could do when they entered cells. And I was obsessed by two central questions. And I make a point of this because I think one of the avenues to success in science is having a persistent and determined, even passionate focus on a couple of central questions. And central questions for me for the next 20-some years were how do these viruses multiply and how do they cause cancer? And while time doesn't permit my getting into the details here, I just point out that understanding how retroviruses multiply required thinking about how they copy the RNA version of their genome into DNA, a process called reverse transcription, how they integrate the DNA copy of their genome, what's called the provirus, into host chromosomes, how they express that DNA-based information from the chromosomes in which the DNA has been inserted, gene expression, how they regulate it, how they make proteins that get assembled, encapsulate new copies of the RNA genome, and then enter new cells. Perhaps more relevant to our discussion today were the kinds of things that my colleagues and I worked on to try to understand how these viruses cause cancer, what kinds of genes do they carry, and importantly, where do those genes come from? In the case of the Rous sarcoma virus, as again I'll say more about tomorrow, we discovered that the viral gene that makes that virus able to cause a sarcoma comes from a normal cellular gene. And that normal cellular gene, as it turns out, becomes one of a large class of genes that can undergo mutations that affect not just viral cancers in animals, but non-viral cancers in human beings. But we also learned that retroviruses can work by inserting their proviruses near such cellular precursors to cancer genes through proviral insertion and by other kinds of mutations. And I'll say again more about that tomorrow. But with time, I began to think as a scientist growing in seniority and rank at UCSF about some of the social attributes of what it means to be a scientist, how science is actually done through partnerships and teamwork, and institutional collaborations, how science can be made more effective by being more open and less secret, and how science can be influenced through public service. And I'm going to say very little about the science itself and quite a bit about how those ideas about how science is actually carried out throughout the rest of this talk before we drift into questions and panel discussions. Vijay has already introduced Mike Bishop and actually coincidentally used the very same quotation in describing the nature of our relationship. Mike and I hit it off very well from the beginning. He too went to a small college, I would argue a little less prestigious college, Gettysburg College, where he majored in history and studied chemistry, went to Harvard Medical School, place I was, as Vijay mentioned, unable to gain admission, but Mike slipped right in. And he and I had and have a shared interest in literature and history, and the love of language that has been essential to a very long time relationship. Partnership is not only the only human interaction that's important in doing science. It's also development of teams. I just pull out one picture from a particularly poignant moment in the history of the Bishop Varmus lab at UCSF when one of our colleagues was dying of leukemia, and that party occurred under these circumstances in the mid-'80s. But Mike and I are seen here, both on the left upper panel showing running a Cold Spring Harbor meeting on retroviruses, and later when our labs actually had drifted apart, but continued at least some level of competition through softball games, shaking hands after one of the teams won the annual Varmus Bishop softball game. But I think when people, even I, think about my days at UCSF, it's pretty clear that at least for the first decade or two that I was there, that I was not necessarily a good community person. I was pretty obsessed with these questions I was trying to answer in the laboratory. I never attended a faculty senate meeting, for example, and that was probably a very wise decision. I never aspired to be a departmental chair. I never joined any committees unless the committees were about selection and nurturing of students. But as time went on, I became more interested in certain kinds of political engagement, not necessarily engagement on the campus itself, but engagement more broadly. And one early manifestation of that was my chairing a committee to name what proved to be HIV, the AIDS virus, arguing with the doyens of the field about what the virus should be called. That's a story in itself, which you can read about now for free in my book, and engaged in trying to obtain more money from the Reagan administration for study of HIV and AIDS. I got engaged, especially after our Nobel Prize was announced, and I was thought to be smart, in fights with the federal government about how much the government should spend on the cost of facilities and maintenance at universities that did research, how we should be adjudicating disputes over misconduct in science, how we should provide better funding for young investigators. Indeed, a group of us at UCSF who were senior scientists at that point, Mike, my colleague, Bruce Albert, shown on your left here, who was about to become the president of the National Academy of Sciences, and a distinguished colleague of ours who's bending over in this picture, named Mark Kirshner, who many of you may know, wrote a letter to the incoming administration in late 1992 after Bill Clinton had been elected president, saying to the president, we recommend 15% annual increases in the NIH budget. And none of us thinking that any of us had enough firepower to make any of our 10 demands on the new presidency reality. But I think I had underestimated what it meant to have a change in status, having won a Nobel Prize and being willing to engage politically, led not too long thereafter to an appointment by Bill Clinton to become the NIH director. And this shows me arriving by bicycle at the NIH. One of the emblematic features of building one at the NIH where the director sits is the recollection that this NIH campus was dedicated by Franklin D. Roosevelt, one of the great heroes of American history, only a few years before, 1940. And his words on those occasions have often stirred my own ambitions to try to do good for the American public. When he spoke from just before his third election to the presidency, saying that the country can only be a strong country if it's a healthy country, as well as a country that has great military might. This was, of course, just a couple of years before the U.S. entered World War II. I want to say a few things about what life was like being director of NIH and dealing with the kinds of things I had not been doing as a faculty member at UCSF. And perhaps good to start with a couple of the high points. On the left is a picture taken in the Oval Office of the White House when the most famous scientist in the world today, Tony Fauci, not quite as famous then, and I'm the guy with the bald spot down in the lower left, we're explaining to Al Gore and Bill Clinton how the AIDS virus worked, why it was so hard to treat AIDS, why we should have a vaccine research center on the NIH campus, which is the source of much of the source of the technology that led to the messenger RNA vaccines that have been so prominent during the coronavirus pandemic. So this was an important moment. Bill Clinton having responded to our suggestions that he get the money to allow us to build a vaccine research center that could be a testing ground for the AIDS vaccines, which we still don't have, but could also be a place where other kinds of vaccines, like the coronavirus vaccine, which we could not then have anticipated, might be generated. On your right is a picture of me sitting in the First Lady's box when Bill Clinton was making the case to Congress during his State of the Union message in 1998 that NIH should experience a large increase in its funding. And this was an unusual moment for me to be on national television sitting next to the First Lady on a day in the middle of the scandal that brought about the impeachment of President Clinton. So there were a lot of things that were involved in running the NIH. It wasn't all the kind of excitement that I've just shown you. A lot of things that you might consider to be routine, like building an annual budget. But the way in which that was done was exciting for me. I was asking the heads of the 20-some institutes and centers of NIH to give me some new ideas, and then I try to give them some new money. But we managed to generate enough excitement about what the NIH was doing that before my term ended, we had encouraged Congress to double the NIH budget over 5 years, which was an important moment in the history of medical science in the U.S. Some of the routine stuff involved building new buildings, which I always enjoyed, trying to recruit new talent to the NIH, defending the agency when it was under attack, especially when there were the occasional scandals or opponents of the NIH rising to try to obstruct its efforts to make scientific progress. Some of the things that I found both rewarding and surprising involved making new kinds of policies about what I argued to be the important goal of engaging more clearly in conducting science, especially medical science, throughout the globe, providing more access to scientific work through both the building of a digital public library called PubMed Central, which I assume all of you use all the time, at least if you're in the biological sciences, and through open access publishing, which is becoming increasingly important, through strengthening clinical research and the relationship between basic and clinical research activities, through development of policies about embryo research and stem cell research, efforts to make some changes in the organization of the NIH and many other things. Just mentioned two of these in a little more detail. Our time is getting a little short. I want to preserve much time for Q&A. But my interest in publication practices grew out of a conversation that I had in 1998 with a former postdoc named Pat Brown, who's now, I hope, becoming famous even in India for his founding of a company called Impossible Foods, in which he is making meat-like substance out of chemicals grown in yeast to allow people to desist from eating the products of animals, but without having to grow those animals, because the stuff tastes like meat. It's great. And he pointed out to me that the physicists of the world and astrophysicists and computational scientists were sharing their work through preprints on a website called Archive that still is very active, and that it was time for biologists to share their work in a similar way. And that meant a major change in the culture of science as well as the business of publication, and illustrated here by a picture of a monk in his private library as opposed to the New York Public Library, where people all citizens can come and go and borrow books. And that led to the creation of the full text digital library at NIH, PubMed Central, led to the development of open access publishing, as illustrated, as Vijay indicated, PubMed Central, and now, of course, a very large fraction of the world's literature, and increasing fractions as funders begin to understand the importance of ensuring that the work they pay for gets distributed as broadly as possible through open access publishing. Second major cultural and policy theme was developing better ways to work with developing countries in doing science. And one of the ways in which I tried to foster that was by engaging my colleagues throughout the world who were working on malaria at a particularly auspicious time for studying this terrible and widespread disease. And a lot of that was carried out through our engagement with a malaria research and training center that NIH had established in Bamako, Mali, in the mid-90s, and through the establishment that we carried out with the Pasteur Institute of the Multilateral Initiative Against Malaria, an organization that now has thousands of largely African scientists at work on collaborative research, mainly in Africa, but throughout the world. As Vijay indicated, I promised my wife that I would leave the NIH after 6 years and go back and hopefully go to New York. And I was happy to be offered a job running the Memorial Sloan Kettering Cancer Center, which was a leadership position, more localized, more focused on my own scientific work, and led to a lot of pleasurable things. And it gave me a chance to think about how to structure cancer research in one institution, building necessary buildings, recruiting new people, emphasizing the modern aspects of science that were going to be influential and have been very influential, in changing the way we diagnose cancers, treat them in various ways, understand individual cancers in a way that is consistent with what we now call precision medicine, and to nurture, as we did by recruiting my fellow Nobel laureate Jim Allison to Sloan Kettering, and to do more of what we now call precision medicine. But not only were there politics involved in running an institution like this, but I became interested in the late part of that decade in getting more engaged with national politics, and began to help Barack Obama become president by running the one arm of his campaign, the science and technology arm, and I joined some colleagues in chairing the President's Council of Advisors on Science and Technology, and then agreeing in 2010 to run the National Cancer Institute. This is a picture of PCAST. I'm over across the table talking to, presenting the results of a study that President Obama had asked us to do on the pandemic of the day, the influenza epidemic of 2009, 2010, discussing with Obama how we go about making better influenza vaccines. And I'm going to be fairly brief about the National Cancer Institute, but it was an institute with a lot of resources that I think had, to a certain extent, lost its way, not kept up with all the science that was available to be done at that time, and at a time when budgets were constrained. Universities had enlarged their research staff, so budgeting was a serious problem, and competition for positions and for funds became extremely difficult. So this led me to think about how to manage a situation in which the opportunities were great, the financial resources were terribly limited, and people had become extremely competitive with each other for the available resources. And two things were important in my thinking at that time. One, the words of Lord Rutherford, physicist in the UK, who had said to his colleagues in 1910, gentlemen, we've run out of money, it's time to start thinking. And so I devoted a lot of time to bringing people together to talk about new ways to make our money go further. But at that point, I'd also encountered in the Metropolitan Museum in New York a staff that was the property of a bishop in Germany in the 13th century that said that he should use this staff to pull people up with the upper part of the crozier, to give them directions with the middle, and then to spur them on with the lower part. And the ideas of generating new ideas, persuading people to engage them, and then prodding them in some way to carry out the possibilities led to a more general strategy for how to revitalize an institution like the NCI with a $6 billion budget. And one was to think about the discovery platforms that existed, and the NIH had already started the Cancer Genome Atlas that I know many of you here are familiar with, and to use information technology combined with genetic information about cancer to design new kinds of clinical trials. The second issue was to try to think about problems that had proven to be intractable, like developing drugs to treat cancers that had mutations in certain genes, such as the RAS genes. And that initiative, still ongoing, was started at that time. I tried to engage the community in defining problems that were intriguing but seemed too risky to undertake, and that led to an initiative called the Provocative Questions Initiative that was intended to engage people in designing new goals for research and to take risks, because we were going to devote review panels to discuss specifically those grants that were taking on these provocative questions. Then there were ways of designing some new grant programs and to focus on certain problems like the rising incidence of cancer in poor countries. And we built a new Center for Global Health that was intended to engage scientists in poor and middle-income countries in cancer research. And then just to think more broadly about how cancer is conducted and to improve publication practices and the sharing of information in ways that I've already mentioned. So these days, having left the NCI after 5 years, what am I going to do with the rest of my days? Lives are limited by inherent biological properties of all species. Life doesn't go on forever. What am I going to do with the rest of it? And one thing is more teaching, and especially teaching about some of the things we've been talking about, how society and science interact. And for the past several years, I've been giving a course that is called Science and Society, that tries to embody some of the properties of activities I've been talking about. How does science work? What is science as a profession? What are the collisions between those who do science and those who expect to benefit from it? So either at City University, where I taught this course for many years, or now at Columbia, this course is designed to ask why somebody becomes a scientist, to something that I'm familiar with from personal experience. Why do governments pay for this activity? What are the limits of science? When can science say something is proven? How does science influence all kinds of government policies? How science is portrayed in the arts in times that range from the writing of Frankenstein by Mary Shelley to science fiction movies. Who owns science? What are patent policies? Who gets to use its products? How do we deal with the costs of the fruits of scientific knowledge? Finally, the other thing that I've been doing in the last couple of years is a lot more advising and efforts to promote science, and making, as I have done for years, becoming part of scientific advisory groups at many academic institutions and biotech companies, even trying to help New York expand its economic development. But I'll just say a couple of final words on this topic by mentioning some recent stuff I've been doing with the World Health Organization, thanks to the influence of the Director General, Tedros, and one of your Indian colleagues, Soumya Swaminathan, who for 5 years was the head of the new science directorate that Tedros had established when he became head of the WHO about 5 years ago. So Tedros decided he ought to have a group that was providing advice to WHO and to the science directorate, not simply on individual topics like tuberculosis or blood cancers or mental health, but instead a group that was devised to set up studies of broad technologies that could influence global health. And the Science Council that he asked me to chair, composed of about 11 people from around the world, took on as its first topic the subject of genomics, the use of DNA sequencing technologies in particular to understand and detect and treat disease more effectively. And just a few months ago, our Science Council released this report on accelerating access to genomics for global health that assigns to WHO a number of what I consider to be important tasks. That is, efforts to promote the adoption of genomics in essentially all countries in the world, a need that has been illustrated very dramatically through the COVID pandemic, the opportunities for implementation of the things that are needed to carry out genomics, equipment, reagents, talented people, service contracts, the way in which groups within countries, different countries, countries working with commercial suppliers can collaborate effectively, and then new ways to make decisions about and deal with the ethical, legal, and social issues that are germane to genomic activities. And I don't have time to discuss this report in detail. I urge you to have a look at the WHO website and look at these recommendations, which are highly pertinent to a country like India, which has made a lot of progress in its efforts to carry out genomics over the last several years. But you live among other countries that have made much less progress, and you can become regional leaders in developing genomic technologies, not just for health, but for agriculture and aquaculture and legal applications of science. And I urge those of you who are leaders in science in India to take on these charges. So just in conclusion, what I've tried to do in this 45 minutes or so is to tell you something about how in my own life I've tried to balance what I would call the art of science, that actually the actual doing of scientific experiments, with the inevitable political aspects of science, and remain flexible enough to change the emphasis of what I'm doing with my own life day by day, how we attempt to respond to either long-term needs like conquering cancer, to short-term needs like dealing with a pandemic or with an inadequacy of resources, conflicts between satisfying our various desires, and also seizing the opportunities to make progress against disease or other inequities in life, by seizing the opportunities that science offers us to improve our understanding of the world around us and to apply that knowledge to the benefit of humanity. Thank you very much.